that by Saturday. Oh, I did that on Valentine's Day, didn't I? Oops. Get it done before Saturday. Okay, so today we're going to continue on with roots. Tomorrow in lab, you'll have some fun with roots. And one of the things that we'll be thinking about when we look at root structure is when we zoom into the angiosperms, which are the flowering plants, that we do see a decided difference in structure depending on if they're a monocot type angiosperm or a dicot. And here you're seeing a comparison. We looked at it last time where we see the dicot has that cross or plus shape to the central vascular cylinder, and the bigger cells, of course, are the xylem. And remember, xylem's composed of two subtypes. What are the two subtypes? One starts with a T, one starts with a V. Tracheids. What's the other one? Vessels. Okay. So the vessels are the bigger ones. So you can see there's some big vessels, and then there's some smaller tracheids. And then the phloem, with angiosperms, it's the sieve elements that make up the phloem. And we can see they're stained a bluish color. They're much more dainty looking. They just have a primary cell wall. And then we looked at how that's all surrounded by an endodermis. And then right to the inside of the endodermis is another ring of cells called the pericycle, which can do active growth throughout the life of the plant to produce those lateral roots. So dicot plants tend to have a taproot, and then they have those lateral roots coming off due to the pericycle continuing to grow and divide and produce those lateral roots. Now this one here, you can see is a ring structure. So this is more of what the dicot is doing here, where you have that central taproot and the lateral root. I jumped ahead a little bit. Then when we get to the monocot, I think of that as a single ring of vascular tissue, mono one. It's just one big ring of vascular tissue kind of in the center of the root. And the xylem, you can see the big vessels here forming that ring. That's easy to pick out. Those are the vessels of the xylem. And then the phloem is to the outside of that. And again, that dainty blue color. And they can also form lateral roots. Okay, they have an endodermis that surrounds all of that, and then that pericycle right to the inside that give rise to those lateral roots. Okay, so when you look at lab, you will be comparing the structure between a dicot and monocot root. Another thing you should kind of pay attention to is the arrangement of the root tissue, because we need to think about that, especially when I start talking to you about stem tissue. We're going to see there's some definite differences to where the vascular tissue is arranged. So in the root, when you look at root tissue, dicot versus monocot doesn't really matter. The root vascular cylinder, or the vascular tissue, tends to be toward the center of the cross section, okay, the center of the root. And we're going to see with stem tissue, the vascular tissue is further out toward the edge of the cross section. So the stem tissue, you're going to see vascular tissue more out toward the edge, at least with the dicots. So I'll get into that a little bit more. Okay, so let's see. We need to think about what roots are doing a little bit, and then I'm going to come back to this idea when we get all the way to the leaves. We'll pull it all together. But we need to start thinking about this idea of the roots. One of the roots' main job, of course, is to take up water and dissolve minerals from the soil and then move it through the plant body. And so that process is called transpiration, which is the movement of the water through the plant body. And there has been really intense research on how this happens, like how can the water move? And first they had to figure out it was through the xylem. And we know that xylem Cells are dead at maturity, so there's no cytoplasm. So that's good because it allows free movement of water. There's no issue with having to plow through all that cytoplasm and all the organelles. So they can just move through kind of like a straw when you're drinking soda pop through a straw or something like that. And so they came up with all kinds of ideas to test, like how is the water moving? What are the physical forces? At first, they thought maybe water's pushed up the plant. There's some kind of pushing coming from the root area. And then that was quickly discarded after they did some real measurements and they could measure the rate of water moving through the body of the plant. They could measure the diameter of the vessels, frictional forces, all of that. So they took all of that into account. And the best theory they have is the cohesion tension theory. So cohesion is referring to the water. You have water molecules that like to hang out with each other. So cohesion, that's what that's getting at. Okay, so the water molecules they like to bond with each other. Tension is the force. Okay, instead of things being pushed up the plant to the leaves, the water, it's a tension where you actually have the tension force generated in the leaf tissue, in the vein of the leaf tissue. So when we think about tension, we're thinking about that force is actually generated in the leaves. And I can't write the S on there. And so when we look at what's going on, the water is actually pulled up the plant from the leaf tissue. So we have to think about how that's happening a little bit. So when we think about water movement, we're zooming into the xylem, vessels and tracheids in the angiosperms. We think about dissolved minerals in the water. There's also dissolved gases in the water, and that will be brought up. In order for tension to develop, you have to have the stomata open, period. Okay, otherwise this doesn't work. The physicists will tell you that. Okay, so stomata must be open. So if you can imagine the sun coming up in the morning, striking the plant body, the plant wants to do photosynthesis. They open their stomata. As soon as they do that, with their goal of I want to take in CO2 for the photosynthesis, absorb light energy. But as soon as they open their stomata, water vapor starts to leave or exit their leaves. And because of that happening, you'll get a tension force developing on the liquid water in the veins of the leaf. So water has to exit the leaf. It does it in the form of water vapor. But that action causes a tension force on the liquid water molecules that are in the vein. And so those molecules are literally getting pulled up the plant, and that force is actually generated due to the stomata being open and then the tension that immediately develops. So this tension gradient will develop. It's a gradient, which means the tension force is really strong in the leaves comparatively. And then it gets, a little bit weaker as you go down all the way to the roots. So it's a gradient of the force. But as long as that water column in the xylem stays together. So because of all the cohesive forces of the water molecules wanting to hang out with each other and bond with each other, that's a good thing because it makes sure that that water is just this solid column of water all the way from the roots up to the leaves through the xylem. So you can think of it as if water is being sucked up the plant through the xylem. And that's as far as I'm really going to take it in this class. In Plant Phys, those of you who had Plant Phys, we get into a lot more detail than this on the forces being generated. But That is, in a nutshell, what's going on. Took them a while to figure this out, but with all of the measurements that they were taking on the different plants, with all the xylem, what's the diameter of the xylem, what force would be needed to get that kind of water movement rate that they were measuring. And the thing is now, the plant always has to deal with this. As soon as the stomata open, tension force gets set up, and you have water exiting and moving through the plant. They can't stop that. If their stomata is open, they have no way to stop that. The only thing they can do is close their stomata to stop that tension force. The tension force will get relaxed, and the plant won't be losing any water, of course, with the stomata closed.
So it's are the leaves. Is that why, like, I've noticed on some of my plants, at least when there's a new leaf growth, sometimes there's a little water droplet on the very end of it. Is that because like it's just so strong that it's like
Can be like if you've ever, and also like what you just described kind of reminds me of, I've not been in a tropical rainforest, but if any of you have gone there or seen videos of it where you see that steaming jungle effect and you see the water droplets on the leaves. And essentially what's happening is water is exiting the leaf as water vapor, and then it immediately condenses back to liquid. And so that might be happening with, especially a young leaf is growing really fast. So its stomata is going to be open a lot, and so I could see perhaps that happening especially with new leaves. Other questions on the cohesion tension idea? Okay, so another thing to think about, and you kind of got into this last week in lab when you were sampling your soil near the roots and you were trying to isolate some microbes. By the way, I looked at your plates, not all of them, but I was picking through some of them. Julian, I saw what you did with the water faucet too. You'll be interested to see those results. Both Jacqueline and I went, whoa. And then we were laughing as we had to wash our hands after we looked at the plates. So yeah, so everyone should take a look at Julian's plate on that. But anyways, remember you did that serial dilution of the soil and then you plated out the diluted samples, and you're going to see lots of colonies of primarily bacteria, although I did see some plates with fungi on them too. It's just fun to look at them. You should look at your group's plates and anyone else's plates that look interesting. I did see some plates that had Streptomyces bacteria, which is the group of bacteria I tend to work with quite a bit because they hang out with plants all over the world. And so I'm very interested in that group. But so yeah, I think you'll have fun tomorrow when you look at those. And they don't smell great, so just be prepared for that. Streptomyces smell great though. They smell like soil after a rain, that nice, lovely smell of wet soil. But most other bacteria do not smell good. So what I'm thinking about is the rhizosphere. What you were doing really was sampling the rhizosphere when you took that sample of soil, because your pot of your plant probably had a fair bit of roots in there. And so when you think about the rhizosphere, that's a term that's given to think about the area right around the root. And they usually define it as just a few millimeters, really, but I would argue with that because now they find that plants are able to excrete chemicals much further than that into the soil. So I'm not, I think that definition needs to be updated. But the rhizosphere is that region of the soil that is influenced by the plant. That's really the best definition of it. That area of the soil that's influenced by the roots of the plant. Roots are very leaky, we say. They like to, they're constantly exuding and excreting all kinds of chemicals, a variety of chemicals. And they're doing a lot of that on purpose, so to speak, because they want to attract certain microbes to their roots and they want to repel other microbes away from their roots. So plants are not at the mercy of everything around them. They have an active role to play in shaping their environment, which was not really, that was not a thing. People didn't think that just a couple decades ago, or even a decade ago, they were catching on. But anyways, this idea that, for a long time, plants just sit there and get impacted by everything else. And that's totally not true. Plants are very dynamic and they're constantly assessing, they're constantly interacting, they're constantly communicating with all the microbes, especially the good microbes that they're trying to recruit and help them. And they're constantly fending off the bad ones. There's all kinds of microbes in the soil. And that's another thing when you look at your plates tomorrow, please realize and appreciate that you're seeing a tiny sliver of the microbes that really hang out with your plant roots. That recipe that we gave you, was it nutrient agar? That nutrient agar recipe, a lot of the microbes in that soil aren't going to like that. And so they won't grow on those plates. So you're seeing just a tiny slice of the pie, but that's okay. We all know that as microbiologists, when we look at those plates, we know that there's far more still that didn't get grown up on the plate. So it's a very dynamic area. There's a lot of research going into this more than ever because of so many, especially in the agricultural world, because of a lot of pesticides and herbicides and nasty chemicals are getting banned. And so they have to figure out what else can we do to help the plant survive. And this idea of using beneficial microbes to help the plant defend itself and protect itself is a big topic right now in the agricultural industry. So we can't ignore the microbes. That's why I wanted you to do this experiment to let you think about this idea that your plant that you're growing is surrounded by millions of microbes and lots of communication is going on between the plant and all those microbes. Okay. So roots are very complex. They often, the normal person doesn't probably think about roots very much because you don't see them. They're buried in the soil and it's hard to really think about them when you don't see them every day. But there's a lot that they do for the plant. Certainly, we can use them for vegetative reproduction. You can, from that tissue, you can clone. You can get roots in the spring sending up new shoots as the temperatures warm up. That's a very common strategy. We know that roots can be used for storage. If you think about carrot taproots, for example, all that lovely nutrition in that taproot. Their roots are always seeking out and foraging in the soil as they continue to grow. They can sense when there's water near them and grow toward it, for example. When they come across a patch of high nutrient soil, they'll slow down their growth and kind of linger. And so their root behavior is very much like you think about with, the fuzzy big animals like bears or something that come along a raspberry patch and sit down to eat. Roots do that too. When they come across a luscious patch of nutrients in the soil, they'll slow down and really take up those nutrients. And then they'll continue to grow after that gets depleted. Parasitic plants are famous for also tapping into other plants. And so we often see that happening too. We'll talk a little bit about those, but parasitic roots, parasitic plants around here, Monotropa genus. The nickname is ghost pipe. We do have that in our woods here, and when it grows, it looks like a fungus because it's all white. Very pretty plant. Does anyone know what I'm talking about? Little plant. They kind of grow in clusters. They do look like mushrooms, but they are a parasitic plant and they tap into roots near them and they're siphoning off nutrients. They can't do any photosynthesis themselves. So that's one good example that we have around here. Aeration. If you are a plant, not around here so much, but if you're a mangrove tree, in the mangroves of off the coast of Florida, for example, I went and did a walking tour of the mangrove plantations there, or I don't know if you call them plantations or what, but anyways, it was fascinating to see all the snorkel roots. So at low tide, some of their roots will poke up. You can see them very obviously, and their tissue is uh, has a lot of air pockets in the roots. So they've shifted their morphology to deal with pretty much being constantly submerged except for low tide. And so they make their roots very airy so that they can more easily have oxygen flow into their roots because, root tissue is alive, it needs oxygen. And if they're surrounded by so much water, that can be a little tricky. So that fancy airy tissue is called parenchyma. Of course, you had to give it a special name and really hard to pronounce. Parenchyma tissue is that name given to roots that will have like air pockets in their tissue. And you can see this even happening with plants that don't live in mangrove swamps. For example, they've done some nice studies with when agricultural fields get flooded and stay flooded due to a severe rain event. And they can show that new roots that are growing in that submerged waterlogged soil will form parenchyma tissue, even though that's not really what they do on a normal basis. And so even plants not adapted to that really wet environment, they can change their root architecture, on the fly in response to that flooding event. Roots, the movement idea, you can see roots, of course, they grow, they move through growing. They definitely are growing constantly. So you have that kind of movement, but then there's some very interesting other types of movement with a very definite kind of different purpose called contractile roots. And so as roots are growing and they're trying to, you have to imagine a really young seedling now. And as the roots are developing, they will actually kind of contract and expand, contract and expand. And as they do that, it helps kind of grab down into the soil better and kind of get them deeper, the root system deeper into the soil so that it really stabilizes the plant really nicely. And it's just a simple wetting drying action that's going on that helps them contract and relax, contract and relax. And then there's some roots that will kind of do a corkscrew movement down into the soil. Again, if you think about a corkscrew, that's a good architecture in order to get deeper down into the soil and make the root system a little bit more secure. So that's a list. Here's some pictures of what we were just talking about. You have all kinds of storage that we take advantage of. I believe you're going to be looking at parsnip roots this week. Parsnip roots, but you can see there's cassava. Ignore the bananas. I couldn't find a picture without bananas, so ignore those. But cassava are here, these two lighter piles of roots, and then taro are the darker ones. So we don't have cassava, or some people say cassava. I see cassava, but this is a tropical or subtropical root of huge importance for feeding people in that area of the world. It's cheap to grow. It's very prolific. It has good yield if you know how to grow them. And so it is a staple root crop for people in the tropical areas. I don't know that I've ever seen cassava in a store around here. Has anyone? For sale? Meyer or anything? I have not seen one. I think I have seen taro though. Taro roots. Kind of weird that they figured out cassava is good for you because it can kill you if you don't cook it right. Raw cassava, do not eat it. So you have to cook it a very particular way to get the toxins out, and then it's safe. Who figured that out? I don't know. I'd hate to be the person experimenting with that one. Okay. Look at, that's a manroot. Look at how huge that is. So that's main function is water storage. You can see the soil is pretty sandy. So living in arid regions, they have to have a way to safely sock away some water. And if you do it underground, there's less risk of an animal coming along and trying to get at it. Here are those snorkel roots we were talking about in the mangroves, where at low tide, you can see that lower picture, the roots stick up like that. And so they can suck up all kinds of oxygen. And then Sir David Attenborough says they hold their breath at high tide, and at low tide, they breathe. So Some people call them pneumatophores. That's another term that they've given it. But that's where we're thinking about that really airy tissue, lots of air pockets in the tissue. And there's some of the snorkel roots poking through there, low tide. Has anyone seen the mangrove swamps? If you get down to Florida on the coast, I encourage you to do that. The one I went on had a nice boardwalk and everything. It was really easy to walk through there and it was really well done. You'll also see and think about Elodea. Some people say Elodia. I say Elodea. So this is an aquatic plant. You'll see prepared slides of Elodea in lab, and then we're hoping we can extract our Elodea tank. Looks pretty sad. We saw like two tiny Elodea plants, so hopefully we can have those to show. But they're an aquatic plant. They're around here. You can find them in especially streams around here in the non-winter time period. And so they're pretty common around here, but they don't have a extravagant root system because they're pretty much submerged. Most of their body is submerged, and so they can just kind of absorb it through their whole plant body. So when you look for xylem tissue, it's hard to even, it's like, where's the xylem? You know, that's the vascular tissue in the center there, but there's not a whole lot going on. So everything's just really quite minimal because they can absorb so much of it just through their body. Orchids, we're going to have hopefully at least one nice orchid for you to look at. Oh, that's not an orchid, that's a corn. Here's the orchid. So the orchid's on the right and corn on the left. So most orchids, it's kind of weird because if you live in this part of the world, the only orchids we see really here are the ones that are growing in the soil of the forest floor. And that's what you tend to think about if you are from the Midwest. But most orchids are actually living on branches of trees in the tropical areas of the world. So the vast majority of orchids live in the tropics and they like to hang out on branches of trees. They don't harm the tree. They're called epiphytes when they do that. Any plant that lives on another plant is called an epiphyte, Epiphytes. Epiphyte. Epiphytes, generally, when you say that word, you tend to think about the ones like orchids that are growing on the branches of trees. Their roots are dangling. So their roots are actually capable of photosynthesis. They green up. They have a pretty thick multiple layers of epidermal cells to help retain water. So you'll look at that under the microscope and hopefully we'll have some nice aerial roots for you to look at as well. And then if you've ever walked through a corn field of active corn plants growing and look down, that's what you would see, these lovely prop roots. And so if you have any architectural leanings and think like an architect, that's actually a really good strategy to make sure your stem doesn't blow over with heavy wind or if it gets brushed by an animal. Those prop roots really can help. Here's some pretty extreme prop roots. This is a screw pine tree, which doesn't live around here, but pretty dramatic. Again, helping to widen the base so that a lot of tropical soils don't have the greatest, the usable soil for the plant is pretty shallow. So when you get beyond that shallow band of good soil, the soil beneath that's not the greatest in terms of water and mineral availability. And so a lot of the root systems of tropical plants are pretty shallow. They don't go super deep. So it kind of makes sense that they might beef it up here above ground too to help with keeping the stem and branches and leaves stable. There's an orchid vanilla. So that's the vanilla orchid. We actually get vanilla. It's a vining orchid. This one doesn't really live on the branches of trees. It vines around tree trunks and not harming it. It's not strangling it or anything. It's not like a strangler fig plant. But you can see the roots are dangling. Here is another, my friend Kim sent a picture of her orchid blooming in the middle of winter in her house, but you can see really fat aerial roots here. Very healthy, happy plant. Kim was the one who used to own a flower shop, so she used to tell me about what it was like, especially with keeping your plants and cut flowers healthy. And not keeping ethylene around your plants in a flower shop. That would not be good. Why would ethylene not be good in a flower shop? What does ethylene do? Remember? Remember that picture of those flowers?
Drops the leaves and
Yeah, too much ethylene around cut flowers and the flowers die, plants die. It looks really sad. So, if you do purchase any flowers or for Valentine's Day or whatever, always use that packet that they provide with you. That should, if it's a good flower shop, they should get the packets that have the chemicals that absorb ethylene. And so ethylene won't be around your bouquet and your flowers will stay fresher longer. Ever see a banyan tree up close and personal? So that when I was in Florida, I went there for a microbiology conference actually at that time. And so that's when I got to see the mangrove swamps. So even though I was there for microbiology, I was doing all the botany stuff outside. So we saw the mangrove swamps and then incredible banyan trees. Those are roots. They grow from the branches down, 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 in the air like that, all the way down to the soil and then plug into the soil. They can get extensive and massive. It's pretty amazing. I'd never seen such a thing in real life until I went to Florida. So. And then there's the idea of some adventitious roots that come off of primarily, if you think about vining plants coming off the vine or the stem. Here, this is poison ivy that's latched onto a tree trunk. And I hope you all know how to identify poison ivy by now. Or if you don't know, do you have like Google Lens on your phone? Does that ring a bell? Google Lens? I use that a lot when I'm out hiking and I don't know what a plant is. I'll take a picture and ask Google Lens to identify it. So be really careful with that. My one grad student, Scott, he worked outside in summer jobs a lot where poison ivy was pretty common and he was highly allergic to it. I mean, he'd overreact to it. So he actually desensitized himself by exposing himself to super diluted poison ivy, like crushed leaves. And then he'd dilute it and expose them, like put that on his skin. It's kind of like homeopathy. And he did that and then kept building up the concentration to the point where he could go get exposed to poison ivy and he barely reacted to it. So that's pretty amazing. He's the only person I know of who actually successfully did that. Yes.
I feel like I've heard that more you get exposed to it, the easy, yeah.
The worse you react, yeah. And that's probably was his problem because it seemed like his summer job, every summer, he kept going back to that job, in the summers. And it just kept getting worse and worse. And so he read about this idea and I mean, it's not just with poison ivy. There's other chemicals you can do that with, but I don't know a whole lot about it myself.
Right.
Yeah, but it's something to maybe research a little bit if you're concerned about that. I've never had poison ivy myself, but I've seen people be pretty miserable from it. The worst thing I've done in the woods is get ticks all over me. Anyone here like ticks? I can handle almost any insect. Snakes, spiders, ticks, no. Kim, who has that beautiful orchid, I went to her house and we walked on a 2-track, it was just in the woods. 2-track high tick season. I went back to her house, plucked 15 ticks off me. And then I had to drive 45 minutes home. When I got home, I found 7 more. So that freaked me out. So I'm like, okay, now my Jeep is full of ticks and ugh. So anyways, poison ivy. I don't know. I might take that over ticks. I don't know. Never had it, so I might be a little too. Anyways, so area roots again, adventitious roots, Boston ivy. If you've seen some beautiful ivy plants growing, especially on old brick buildings, they tend not to rip those off because it looks so stately, hopefully. And you can see they're pretty well secured there with those adventitious roots on the brick wall. And then look at these. So when you have prop roots that really go into overdrive, then you start calling them buttress roots. And so again, it's the same basic idea. It's usually tropical plants. And they do that to especially bigger, taller trees. So this is a fig tree. And you can see with that shallow soil, their roots aren't very deep into the soil. So they compensate by doing this kind of a architecture. Look at that one. Pretty amazing. Okay, contractile roots, this drawing is trying to get that across. How they're trying to show like the roots kind of crinkle up a little bit. So they're contracting and then relaxing, and it pulls the root system deeper down into the soil. Mistletoe, parasitic. Actually, it's a hemiparasite, which means a hemiparasite can do photosynthesis. They're just too lazy to do it. So they'll parasitize some other plant. Here you can see this is a picture taken, I think, in Australia, but we certainly have mistletoe in America. And you can see this poor tree is well colonized by mistletoe plants up in the branches. So their root systems have actually plugged in to the branch, reached the vascular tissue, and is sucking away nutrients instead of doing photosynthesis. So not a very nice plant. It's kind of funny that we use it as a, mistletoe at Christmas time. You're supposed to kiss under the mistletoe, and yet it's a parasitic plant. So it's got an interesting folklore though, to the mistletoe. It's been a revered plant for many, many centuries, primarily because it stays green in the winter. So it was a hopeful sign to people that, things are still living. And apparently if they would have a treaty with warring people, if people were fighting with each other, if they met under the tree that has mistletoe, it was a truce. They had to call a truce. Anyways, it's lots of folklore about it. This is that Monotropa plant, the ghost pipe. Kind of looks like a pipe that, you smoke tobacco out of. It kind of looks like that. And so you can see no chlorophyll in that plant. And they do look like mushrooms, but their roots are tapping into neighboring roots. Actually, what they discovered is the roots are colonized by mycorrhizal fungi, which then tap into neighboring roots. That was actually a master's student's thesis. That's what he did for his thesis project. He discovered how the roots are colonized by the mycorrhizal fungi, and the fungi are tapping into other roots nearby. So it's like a mycorrhizal bridge from plant to the parasite. And then if you are a corn grower, you don't want this plant, even though it looks really cute, very pretty. It is a Striga genus of plant, and that's a corn plant. And so here on the very bottom of this right picture, that's the root of Striga, and it's plugging in to the corn stem and is going to get to the vascular tissue and start sucking away water and sugars. We do not want this because this weakens the corn and makes the corn grow more slowly. They're trying all kinds of strategies to stop this. And that Hostoria is a name they give to any modified, in this case, it's a modified root that's being very aggressive at plugging in and getting into the corn. And then they're trying to do a lot of different things. They're trying to get crop plants modified to have resistance genes. That's not going so well, last I heard. And then of course, all the pesticides they're trying to, or the herbicides, I should say, they're trying to spray on Striga. That's not going so well. They're trying to use hormone sprays. That has actually been not so bad. That what they do is before they plant the corn in the spring, they'll spray the soil to get Striga to germinate. So they'll probably it's gibberellic acid, I'm guessing, that they're spraying. And then once the seeds start to germinate, they'll, unfortunately, they'll use a pretty nasty herbicide to get rid of it. And so basically what they're trying to do is get rid of the so-called seed bank. So all the seeds that fell at the end of the last growing season and are lurking in the soil there, they're trying to get them to germinate and then get rid of them. It's a big problem. Millions of dollars of loss due to that one little parasitic plant. Root systems are huge. That's my brother-in-law at his brother's place, and it was a big windstorm here in it's up by Forestville Road area, and one of their huge trees fell over. And that's not even the complete root system. That was just part of it that came out. So as you're walking through the woods, appreciate that most trees that you're looking at above ground, the biomass of that tree is bigger below ground. So usually root systems are bigger than the above ground. Not all the time, but it's pretty common that root systems, there's more biomass below ground than what you see above ground. So it's just hard to think about it when you don't see roots on a regular basis. Although Michigan Tech University, they have a rhizotron area, I think up in the forestry building. I have yet to see it, but they built it after I graduated and left. But it's basically a tunnel system in their building that they put plexiglass instead of, concrete walls. And then they grow plants and they watch, and they have these high power fancy microscopes right up to the plexiglass. And they use that to look at root growth over time. So that's one of the best ways that we have to try to figure out root systems is using they call it a rhizotron. So lots of money to build that, but at least it gets them some good data. Okay, questions on roots? All right, let's see. I want to get into let's get into stems while we have a few minutes here. So we're working our way up the plant. Okay, so now we need to think about stems and this idea of, okay, we have this root system and then we have to get it to blend into the stem. And that used to confuse me when I was first learning anatomy of the plant, because you're going to learn that stem tissue, when you look at their vascular arrangement, it's different compared to what we see in the root tissue. So for example, this one on the left, that's a dicot stem. And you can see there's a ring of vascular tissue. Whoops, come on now. There's a ring of vascular tissue. So all of this is vascular tissue, okay? With roots, we just finished roots. With a dicot root, how was the vascular tissue arranged? The cross, yes, in the middle, right? It was, so when we think about root tissue, the vascular tissue was right in the middle. And with a dicot, the xylem was forming that plus or cross shape. So we have to go from that architecture in the root system, and then it's got to morph into this design where you have the vascular tissue in a ring. And so that always used to really bother me until I found a picture that helped explain it. So there's a so-called transition zone where the root and the stem meet. And you can see that the artist is trying to show how you can transition from that plus shape in the root of how the vascular tissue is in the center of the root. You got that plus shape to the xylem and then the phloem and all the corners there. And then you can see how you have that transition zone to where you get finally that ring of vascular tissue. And so it's not a very big zone, but it's enough room there as you transition from roots to stem to get that coherent flow and transition from a central vascular cylinder like that with that plus shape to the ring shape. Also, notice with the root, the vascular tissue's dead center of the cross section. Here, the vascular tissue is closer to the perimeter of the stem. And again, if you think like an architect, that's pretty good because if you build a high rise, you don't put your steel beams dead center of the skyscraper. You put your beams out on the edge or the perimeter. And so this allows, remember now, xylem has thick secondary cell walls. So not only is the xylem, it's important for water and mineral uptake, but it's also good for structural stability. And so if you think like an architect, this is a good design. So you can understand why they went through the bother of doing this transition zone and getting their vascular tissue in the stem a little more close to the perimeter, because that definitely does add stability to the stem. Does that make sense? Okay. So we're looking at vascular tissue. So the bigger kind of purpley red cells that you can see, that's the xylem. Okay, so it's kind of clusters of vascular tissue, but overall it forms this ring, all the way around this ring of vascular tissue. And then the phloem is that more delicate kind of light blue right to the outside of the xylem, the delicate light blue area. And then these are phloem fibers that you see kind of capping it and protecting it. Those are phloem fibers. So you have the xylem and phloem near each other again. It's just instead of doing that plus design like we see in a dicot root, the dicot stem converts up into a ring. And then the monocot does something even weirder, does that. So all those little dots like that, that's the vascular tissue. It's just scattered through the whole stem. And that has serious consequences for how a monocot can grow. So we'll talk about that next time. Thank you for your patience. We'll stop there and continue on Friday.